Today, I think I know I have a long name in the abstract. So for a good memory, I have a logo and a short name for my project called FENRIR. So here, you see, as reflected by the title, it's enhancer functional networks. So I'm trying to understand the enhancer functions from network aspect. So let's talk. When people talk about the enhancer functions, usually we look at the gene first, say which target gene this enhancer is associating with, which target gene is linking with this enhancer, no matter proximal or distal. So let's say if the gene is relevant to a specific cellular process like cell cycling, it's a heart gene, or it's showing up in any cancer study, or it's linked with any, well, a more challenging disease like autism, then we say, okay, kind of this enhancer can be functioning in the relative context. But now I think our knowledge is growing, but for any specific function, tissue, or disease, probably we only know a couple hundred of disease genes at most. Our knowledge is growing, but seems currently it's a bottleneck to interpret the enhancer functions, because I think this morning somebody mentioned that there are over like 1 million enhancer elements. To understand their functions, we need some prediction work based on what we already know. Probably after 10 years, we know most of them, but currently we have the bottleneck. We need to think about, do we really need to understand gene function first, then enhancers? So my work is trying to understand the enhancer function based on existing knowledge, rather jump out of the bottleneck of the gene functions. So let's take a disease for example, because so many people are interested in disease study. If we say an enhancer can be associated with a disease, say it's regulating a disease gene, or from GWAS study, you found a very significant GWAS SNP within the enhancer body. But every year there are some GWAS study published, and they add more individuals. So that means currently, if it's a rare mutation, we observe rare mutation in the future, it can be GWAS significant. All these rare mutation can come from any specific group of patients. We do the whole genome sequencing, we identify variants there. They can be causal mutation, but they are rare. But to be honest, for majority of enhancers, we don't have any direct evidence to say this enhancer is associated with any disease or cellular functions. So that means we need some prediction work. So my work is trying to think about if we can build any relationship between enhancer we already know associated with the disease to those enhancers lacking enough observations. So we proposed this hypothesis called enhancer functional interactions. We thought if an enhancer have a functional relationship with another one, it should share the following 3 features. There are some other features relevant, but this is the 3 things we picked out. They are really contributing to the story. The first one is they are interacting in the 3D genome. So when people analyze Hi-C or ChairPad data, well, most of them, they are interested in the enhancer gene interactions. But when you analyze the data, you actually observe a lot of interaction between enhancer and enhancers. And some people, they also observe interactions between promoter and promoters. They are physical interactions, so they are also significant. So they have their own biological meanings. And also, we thought transcription factor is also important, because some transcription factor, they are also called the enhancer activator. If 2 enhancers, they are regulated by the same set of transcription factor, we saw that they can be active in the same condition. So assuming they may involve in similar functions, or they have some like a correlated activity. Actually, the first 2 things has been used in some way to identify called enhancer modules. People, they then interpret the gene functions of the enhancer modules. So here, as our lab, as an Olga's group, we work for a long time for the gene function predictions. So we have the prediction between pairwise genes, say, if those 2 genes, they are involved in similar or the same cellular processes. So let's say if we have added these 3 evidence together, and we found that this enhancer, they are lacking direct evidence, but it's also interacting frequently with those enhancers that are already associated with disease. Then if you ask me if this enhancer can be disease associated, I can tell you it's possible. It's still possible. It's not like a binary, like a true or false, but I can give you a probability, say, likely this enhancer can be functioning in this disease. And this is, I developed a method to predict this enhancer functional interactions. So it's a Bayesian inference. Bayesian inference means it gives me enough flexibility to study the whole network piece by piece, to jump out of the dependency of signals. So this is a cartoon-wise layout of enhancer networks. In the top layer, we assume there are functional interactions between enhancers. And in the downstream, they are regulating their target gene. Not necessarily like one enhancer regulating one gene, it can be multiple genes. And for the top layer, our observation will be like transcription factor binding signals. And also we observe the enhancer physical interactions from those chair-pad or high-C data. And coming to the downstream, we also have the enhancer gene interactions. It tells me which enhancer can be associated with which target gene. It's not a random pairing. And sometimes the nearest target gene may not be picked up. And also between pairwise genes, we have the functional prediction between them. And then we define a network probability. So based on the observations on the right side, we try to predict the strength of pairwise enhancer functional interactions. As it's a Bayesian inference, that means of giving the positive probability, I can extend it from using the prior and the likelihood functions. And then I can study piece by piece, means I propose a enhancer network structure. And then I study if they are tightly interacting with each other in the 3D genome, if they share like a similar transcription factor binding signals, and if their target genes are involved in the same or similar cellular processes. If everything is high, that means we have a good guess, means this network structure has a high positive probability. But the true thing is, even though we have these observations, we still have like in my case, it's like 50,000 enhancers to be studied. And we know the enhancers are highly spatial correlated. It means in the 3D genome, there's a domain called TAD, or topological associating domains. So that's why I use a sampling technique to solve this problem. So I sample the enhancer network in a local way to fully capture their local structure in the TIDE. And I also specifically designed the jump function to make sure I think the local model can jump out of the TIDE. It means the enhancer from different TIDE, even from different chromosomes, they can be paired together. Because when we study genes, we never think genes in the same chromosome, they always work together. From chromosome 1, from chromosome even X, genes can still be involved in the same cellular process. The same applies to enhancers. And if you can think about this as something as a spider climbing on the whole space of enhancers. If it stays somewhere for a long time, means that there's a signal. If it jumps quickly, means that there's no signal. So if we add up all the sample subnetwork together, we can get a weighted network. So for each pair of enhancers, we get a probability, say how likely they can be functionally related to each other. And when we get the network, the first thing is if my network is making sense, means if it captures tissue specificity, if those interactions, they meet some expectation of earlier studies. It's just not like a random pairing stuff together. So here I studied many tissues. I think definitely I take brain as a prior because it's useful in my later disease analysis. But we use super enhancers, EQTLs, and ERA, since those signals, they have different aspects of tissue specificity, and they all contribute to the enhancer story. And it seems in all studies, I see the signal differences compared to the background signals, all between like a brain tissues versus non-brain tissues. So there's some signal, we capture some signal. And the goal is not just the network, because the network is a whole global picture. The goal is still to identify disease enhancer associations. So we have a second machine learning method. Here, this is our network. And if I have a good quality GWAS study, I can label those enhancers with significant GWAS names as positive, and label the enhancer without significant GWAS names as negative. But you know, if we have so many enhancers, a lot of them actually don't have any GWAS overlapping. So I treat it as a black box. So then we using this machine learning framework, we check if this enhancer is interacting with other enhancers more likely, more similarly to the negative one, or more similarly to the positive one. Then we assign probability for each enhancer, denoting how likely it's associated with a disease. And for some disease like autism, I think the GWAS capture some signal, but can't capture that enough. Means this is a highly heterogeneous disease. So we also provide a function means using the disease gene to label those enhancers. If you have the disease gene with autism, we can label the positive enhancer, and some disease and some genes we know they are relevant to other diseases, but they are not neurological. then it can be used as negative cases. We can still build a classifier to predict all the enhancers. So we build a web server, so user can upload their GWAS or select their disease gene, then get the training model, and then prioritize all the enhancer regarding the disease association. I think it's pretty fast. It's just 2 or 3 minutes you get your results. But fast, that doesn't mean it really works. So the first thing I need to check widely. Let's say we pick up whatever we found from the public resources, say if this my prediction meeting some like a criteria or it's above some expectation. So we compare the first database we check out the disease enhancer. It's a small paper, but it occurred a list of enhancers with existing study linked to a specific disease. And we also checked the OMIM. This everybody may know this database. We checked our top predicted enhancers if their target genes are already labeled as specific disease, labeled for specific disease. And in both works, it seems it's better than the GWAS p-values. And the next one, we pick up a specific disease. It's autism. This is very challenging, and we know the non-coding signals should contribute to this disease because we already carry out a lot of whole genome sequencing data or whole exome sequencing data, but we can't identify all the causal locations. And earlier this year, our group published a paper using a deep learning pipeline to identify some causal mutations in the autism. But in my case, I care more about what's happening in their enhancer regions. So I take the top part of enhancers and check what target gene they are. And if this target gene, they are enriched with any specific cellular processes, which we know they are autism-associated. And also for those enhancers and their target genes, we also check the existing database, say if some other study, they found similar observations as what we have. But until now, everything I talked is on paper, right? It's on public data. Even though you don't use my pipeline, you can still identify some signal similar to this. So our contribution is trying to pinpoint some new enhancers previously not reported, but we show it's relevant to this specific disease. The first thing we check we use the mutation signals from the almost 1,700 families. And we say probound means they are ASD affected children, and the sibling is the children from same family but not affected by ASD. And we check if we found any significant mutation enrichment in our top predicted enhancers. And we do find a significant difference. I will call it significant, even though the p-value is not crazy low, but we do find some difference. And we select several enhancers, say if we can test. The first thing is if this enhancer really carries the regulatory activity. It can be a false positive enhancer for sure, right? And also, if we do observe a mutation here, comparing between probound allele and sibling allele, if we found a differential regulatory activity. So we do the experiments. Let's say this is let's say this healthy allele is not affected, and this is the probound allele with this mutated loci here. We test the 200 base pair around this mutation area, and all of them are falling within the enhancer region. And we do find a number of enhancers, and I just use their target gene to label the enhancer, but actually we test the enhancer region, and we do find the significant difference between the probound allele and the sibling allele. And the enhancer activity is not necessary to be always positive. It can be also negative, regulating the gene. And this is the whole story. And I'm from Flatiron Institute of Simmons Foundation, and we are also affiliated with Princeton University. And this is my advisor, Olga. You may see her in different platforms. And this is the main contributor of this project. And I also want to thank all the lab group members for our internal discussions. And by the way, I think this guy, Chen, I think if you are deep learning folks, you may have heard about his previous work, DeepC. And now he opened a new lab at UT Southwestern Medical Center. So if you are interested or you know somebody interested in working on DeepC or deep learning, feel free to contact him. Thank you.